Hello, everyone. I'm Tiff Bortwood. I'm the Managing Director of Australian Genomics. Welcome to DNA Dialogue, the seminar series where we share new developments in health genomics and have conversations with international leaders in the field. I would like to begin by acknowledging the traditional custodians of country throughout Australia and their connections to land, sea and community. I'm joining you today from the lands of the Wurundjeri Woiwurrung people of the Kulin Nation. I extend my respect to their elders past and present and extend that respect to all First Nations peoples joining us for this webinar today. We do like an interactive webinar, so please feel free to drop any questions that you have throughout the presentation using the Q&A function, and I'll do my best to get through as many of those questions as I can at the end of the presentation. So it's my great pleasure today to welcome Dr. Segun Fatumo, who is an Associate Professor of Genetic Epidemiology and Bioinformatics at the London School of Hygiene and Tropical Medicine in the UK, leading their Uganda research unit. He's also head of the Non-Communicable Disease Genomics and the African Computational Genomics research groups at the London School of Hygiene and Tropical Medicine, Medical Research Council, and the Uganda Virus Research Institute. Segun co-led the first major GWAS of cardiometabolic traits in Africa and led the first GWAS of kidney function in continental African populations. He is strongly committed to increasing diversity in genomic studies and was recently awarded the prestigious MRC Impact Prize for advocating for the inclusion of Africa in genomic research and championing genetic risk prediction of complex diseases in Africa. Absolutely thrilled he has joined us at this very unfriendly hour for the UK to share with us his work and also broader progress in the area of genomics with African populations. Segun, thank you so much for joining us.
Thank you very much for that introduction. I'm going to start by sharing my slide, and I would like to thank the organizers for inviting me to make the presentation today. So good morning or good afternoon, everybody. I'm really pleased to be here today and to make my presentation. So this work is titled to Unlocking the African Genomes for Precision Medicine. As introduced, my name is Segun Fatumo. I'm an associate professor at the Department of Non-Communicable Disease at the London School of Hygiene and Tropical Medicine in the UK. I'm also at the MRC in Uganda, where I lead the NCD Genomics and the African Computational Genomic Group in Uganda. So I'd like to start with this. Medicine, as you know, has evolved over the past decade. In the past, medicine was practiced according to signs and symptoms that are presented by patients and was solely based on the individual knowledge. Most of the time, it's always the knowledge of the doctor or the physician, and with the intention to provide some solution. So currently, at the present, like we know, medicine is based on evidence that is produced from scientific research, including clinical trial. And you will have heard about a lot of clinical trial during the COVID for the vaccine. In the future, which is going to be my interest for the rest of the presentation, so medicine will be practiced according to algorithm, so that will take into consideration many factors, including the patient characteristics, such as their genomes, which is very, very important. There will be other consideration like their epigenetics, lifestyle. This is very important for medicine in the future, and that is what I meant by precision medicine. To explain a little bit more, precision medicine is very important because we are all different. So should our treatment also should be different. On the left, where I put my mouse, it's an example of a patient population. What you will see on the left here is that we are all very, very different. We have so many characteristics. Some are black, some are white, some are different color. We have also, our genomes are not also perfectly the same. So you can see the patient population we are very different. So some are disabled, some are not disabled. So there can be the standard treatment approach is a treatment where everyone is put in the same box, and the same treatment is now applied to everyone. So in such standard approach, so treatments could be effective in only 20% of the target population, and the remaining 80% become a waste. In a tailored approach or a precision approach, you see people are grouped according to different characteristics. What you will see, like you see these people with the same color, it could be people of African ancestry, and they have similar characteristics, and they apply treatment A. The second group of people could be people of Asian ancestry. They have similar characteristics. They apply treatment B. The third one could be people of, say, South Asian ancestry. They will apply treatment C. And the last one could be people from European ancestry and apply treatment D. So this kind of approach will be more effective than a standard approach. So as you can see on this slide, governments of over a dozen countries have invested heavily in establishing national genomics and medicine initiative. This is how important precision medicine and genomic medicine is. If you look at the map, you see from Saudi Arabia, Qatar, Turkey, Brazil, Japan, Estonia, and Australia, you see United States, United Kingdom, you see a lot of investment into genomic medicine in order to accelerate precision medicine. Very popular is the Australian Genomes, the United States All of Us project, which aims to recruit about 1 million people from the US into the study for precision medicine. You may also be very familiar with the UK Biobank. This has been a well-characterized initiative in the UK for in the last few years. So you see what I'm trying to bring out here, you see this genomic medicine is very important, and that shows in why governments of nations from all over the world are investing heavily in this initiative. So you see Chinese government announced a bold initiative to become a global power in precision medicine with about 9.2 billion for 15 years. So they are aiming to recruit, I think, about 100 million people, doing about 100 million whole genome sequencing by 2013. But what I really want to emphasize on this slide is, if you look at the middle, here, you see the map of Africa. So you don't see any initiative coming out from Africa as a continent. And that's what will be a focus of my presentation today. So Africa, I don't want to make an assumption. Africa is a continent that about 1.4 billion people are living there. And I mean 1.4 billion people. That is a lot of people. So Africa, like you know, is not a country. Africa is a continent that has 54 different countries. So they speak about more than 2,000 different languages, grouped into about 3,000 different ethnic groups. Africa is the world's second largest and the second most populous continent in the world. What might be surprising is that 50% of African population are children less than 18 years old. In Uganda, where I do most of my work, average age in Uganda is less than 16 years old. It's very similar also to what you see in Nigeria and many parts of Africa. By 2050, 40% of what children will be living in Africa. So this shows how important this continent is, and this continent should not be left behind in the coming revolution of genomic medicine. So African genome is very important. There have been several genetic studies that have confirmed the theory that modern women we evolved from Africa and they migrated to Europe, Asia, to reach the Pacific and America. So as you know, African population today about most genetic diversity than other populations in the world. And the genetic diversity that is found in non-African population represent largely only a subset of genetic diversity that is found in Africa. That shows how important the genetic from Africa is. What I really want to emphasize on this slide is within Africa, there's even larger genetic differences within African country than between Africa and European population, or between Africa and East Asian population. So what this means is that South African genomics might be very, very different from the Uganda genomics compared to African and European population. So this genetic diversity has a lot of implications. Number one is that genetic study of African population are crucial to fully understand the modern human evolution and the effect that it has on human disease. So secondly, this may imply that results from genetic study in non-African population, including their clinical relevance, may not be generalizable to African population. So thirdly, this may imply that there are many genetic variants that are yet to be discovered, which may probably play a role in medically important phenotype of African population. And the fourth implication is that this may imply that findings from one genetic study in one subpopulation within Africa may not be applied to another population within Africa. An example is maybe findings from South African population may not be applied to Uganda population. So there is a need for more representative genomic study from all population. There's a drastic lack of diversity in genomics, despite repeated call and warnings. So for example, as of 2016, a study revealed that 81% of participants in genome study, GWAS, were people of European descent, with only 3% from African population. 3 years down the line, in 2019, the proportion of Africa in genomic study declined to 2%. And again, in 2022, in a paper that we published in Nature Medicine, so we show that this proportion of African in genomics further declined to 1.1%. So this suggests that in spite of call for more diversity in genomic study, the gap continues to widen. It is crucial that we need to address the lack of diversity in genomics as a global effort. So what is really important to note is that even the 1.1% of participants of African ancestry in genomic study, which I mentioned just now, are mainly Africans and African American, and the proportion of continental Africa in genomic study is insignificant with respect to the prevailing genomic study. So therefore, if we continue to study a small number of African diaspora population, and we continue to group them as all participants from Africa, we continue to broaden category, we continue to just widen the age disparity, and we will fail to capture the full genetic diversity in African population. So in a recent correspondence that I wrote to Nature, I emphasize this. We emphasize that the mistaken presumption that the genetic of a relatively small diaspora from Africa that is applied to all African population will continue to threaten and widen age disparity. So in this correspondence, I give an example of about the 5,200 North American individuals. And in that study, half of those people were from just a single region in West Africa by their genetics. There are so many common ancestry like Nilo-Sahara speaking people and Kosan Otagadora people. They are extremely underrepresented in genomics. We should remember that Africa is home to some 3,000 ethnic groups, should not be neglected in genomic studies. So here I give an example where genetic data of underrepresented population are available, but neglected for analysis. If you go to conferences, you hear people talking a lot about diversity in our population. So UK Biobank, like I mentioned previously, is well characterized, has about 500,000 individuals there with data available for genetic data and phenotypic data available. So this database also has about 38,000 people that are non-European ancestry individuals. So, but what you find in publication is 93% of publication excluded non-European sample when they do the analysis. So this is a case where data is even available, and they are being excluded for analysis of for study. So we continue to say to talk something and do something that is different. Here's another example of an excellent paper that was published late last year. So this is a GWAS of height where they try to find genetic variant that are associated with height. Height is one of the most common traits and very easy to measure in our population, including the African population. Most GWAS in Africa currently have probably measure height. But what is very concerning in this paper is they aggregate data from all across the world, 54 million people, more than 280 GWAS aggregated, and you don't find data from Africa included in such study. So this is a case where data is available but excluded. So my question is, why exclude indigenous African population even when data are available? So unfortunately, there is a bias in the current global biobank for genomic study. Here, I'm giving another example of where we have to put, when we have to put Africa on the map for genomic study. So this is another great initiative trying to aggregate 23 biobank globally, which make about 2.2 million individuals. And if you look at the map also at the middle here, you see that there's no African that is represented. You see biobank from all over the world be represented in this great initiative. So, but recently that's changed a little bit. If you look at middle now, so you see just one biobank in Africa, which is Uganda Genome Resource, an initiative that I currently lead. So there's a growing number of African genome resource that are capturing the true genetic diversity across Africa. So this resource that's capturing the diversity, they are great value for discovery and genetic prediction that can generalize more broadly across a population. So here on the right, I show a map of where the initiatives are coming from. You probably have heard about the Human Hereditary and Health in Africa, H3 Africa, and you probably have heard about the great, excellent work that have been done in the last 10 years to recruit participants all across Africa. And the other initiative, you see these genomic resources are coming from everywhere in Africa. These are great resources that needs to be used for discovery and genetic prediction. So why is this so important? So one example is the PCSK9 inhibitor. So this led to discovery of a highly effective new class of drug that lower bad cholesterol. This discovery was only possible because some African ancestry individuals were included in a study. So in that study, the discovery paved the way for development of this drug because we had some African people were in the study, and the study found that those African people naturally have some genetic variant that helps to lower cholesterol. So what I want to emphasize on this slide is we probably will not have PCSK9 inhibitor and this drug if those African ancestral individuals have not been included in this study. So this drug is now doing the miracle, not only for Africans, but for everyone all over the world. So I think that we are doing ourselves harms if genomic study is not diversified to include Africans and other underrepresented population. So just recently, last month, there was a paper that was published in Nature, which I was also one of the co-authors. So in this paper, there's a genetic variant in people of African descent that reduce HIV load. Like you know, nearly 39 million people worldwide are still living with HIV. There's no cure, and there's no cure or vaccine for that. So this African-specific variation at a gene called CHG1L is most likely causal gene at that loci. And this is very, very important gene that has the potential for revealing therapeutic targets for HIV. So again, the emphasis here is that this study, including African ancestral individuals, are very important for the global population. So here's also another example of a gene called ApoE1. So ApoE1 has currently there been a new drug targeting ApoE1, which is showing promising results for patients with inherited kidney disease. You might know that people that inherit two ApoE1 mutation are most likely going to develop kidney disease compared to people without the ApoE1 mutation. So this chronic kidney disease is at least 3 times or more, 4 times, even more frequent in Africa compared to developed countries. So with this kind of study coming up and potential drug coming from this, so there's a possibility that this will be a drug that will be effective to help with chronic kidney disease in Africa and African ancestry living all over the world. So today I'm talking to you about some work that we are doing that I'm currently co-leading or leading in Uganda. So there's one called Ugandan Genome Resource. So this is a cohort of about 22,000 people. We've already genotyped about 5,000 people, did whole genome sequencing, an additional 2,000 people. So we plan to expand this initiative to include multi-omics, proteomics, metabolomics, epigenomics, and single-cell genomics. I'm also co-leading an initiative called Malawi Genome Resource, which this is a project that the sample collection was led by colleagues in Malawi, Professor Mia Crampin and others. So this is from a cohort of about 25,000 people collected from rural and urban community. So we have already currently genotyped about 7,000 people. I also plan for expansion. I will talk briefly also about the project that is called the Nigerian 100,000 Genome Project. So all these project initiatives I mentioned, they have strong community engagement. An example is Ugandan Genome Resource, which is from the general population cohort in Uganda. So having strong community engagement help this cohort to flourish. The general population cohort in Uganda has been in existence since 1989. So, and 1989, we've been going back to this community almost yearly to administer questionnaire and collect biosample from them. This has been a very successful cohort for a year. In 2011, we started genetic epidemiology of non-communicable disease. And what I really want to emphasize here is the community engagement that we always engage in the community. So, for example, we have a community advisory board, CAB, that consists of stakeholders in the community, such as pastor, imam, and cultural leader, that help us to shape our study in Uganda. So one emphasis to make is the need for community engagement for a study like this. Community engagement, not just consultation or research announcement, is key for genomic study, especially in indigenous population. It is not good to only show up when we need to collect the sample, like we know. So we in 2019, we published a paper in Cell. That paper represents the largest ever genome study of Africa. So largest ever genome study of Africa was only about 5,000 people that were genotyped from Uganda, an additional 2,000 people that were whole genome sequenced. So at that time, so they were genotyped on 2.5 million Illumina array. So in this study, we also incorporated data from other parts of Africa, including data from Uganda, from Nigeria, from Kenya, South Africa, making a total of about 14,000 individuals across Africa. So like you would expect, the study from this Uganda genome results show evidence of population substructure. We can see different ethnic groups coming out clearly from this PCA. So this can be challenging. It can bring out a confounding for a study and may require some very specialized approach, such as mixed model, to analyze this kind of data. So as you may expect, we found a higher level of genetic diversity in the Ugandan population than it is seen in similar study of European population. Around a quarter of genetic variation identified in Uganda data set have not been discovered before. Specifically, we found that 28.7% of SNP discovered in Uganda whole genome sequencing, only 2,000 samples are not found in the genome aggregation database at that time. Specifically, like you can see where I put the arrow, we identify 9.5 million novel variants that are not present in the 1,000 Genome Project Phase 3, at that time, the African Genome Aggregation Project, and UK 10K. So we demonstrate the value of African genome resource by creating an African unique reference panel using using the 2000 whole genome sequencing, which I mentioned earlier. As you may see, this is a bit small. Our unique imputation reference panel outperformed the existing imputation reference panel in terms of accuracy, especially when you are doing the imputation into East and South African population. So this slide shows the list of novel genetic variants. I mentioned that we did a genome version study, GWAS, in about 34 different traits. So what I really want to emphasize on this slide is the last two columns here. So this column here is the minor allelic frequency in African population. And the last one here is the minor allelic frequency in European population. What I want to emphasize is that some of these variants that we found to be associated with a various cardiometabolic trait are monomorphic in European population. So meaning that they are either absent or very, very rare in European population. So what this means is that this genetic association can only be found in African population because they are not present in the European ancestry individual as shown by our study. So here's an example. So our discovery includes a genetic variant that causes alpha thalassemia to be associated with HbA1c. HbA1c is an indicator for diagnosing diabetes. So this genetic variant is present in about 22% of African and almost absent in the European population. So this particular variant is thought to have become even more frequent among African population because it can prevent severe malaria. So here, our studies suggest that as HbA1c levels are often used to diagnose diabetes globally, it is possible that diabetes may be misdiagnosed in those 22,000 African because of this association that we found against HbA1c. So our study also leveraged a lower linkage disequilibrium in African dataset. The pharmacopeia analysis resulted in a narrow credible interval for most traits. Specifically, we were able to map 16 out of 41 distinct loci to a single SNP in the credible set. We also resolved the previously identified association with HbA1c. So very interesting, here we assess the heritability of all the 34 cardiometabolic traits. So we found that height is less genetically determined in Uganda relative to what you would see in the European population. The narrow sense heritability for eyes was 49% in Uganda, compare that with about estimate of 80% in the European population. In contrast to that, so we found that the heritability estimate for LDL was significantly higher in Uganda, about 54%, compared to about 20% in European ancestry individuals. So collectively, our findings from this small data set show a great potential and value of African genomics data for discovery. So since then, we have been now leveraging regional and global collaboration to improve power for discovery. So within Africa, we collaborated with study within H3 Africa. We are also leveraging data from African American individuals, including the Million Veteran Program, MVP, and also UK Biobank, which I've mentioned earlier. So this is a study that was published in 2021. So this is the GWAS of lipid traits in global population. When it was published, this was the largest ever multi-ancestry GWAS, where they aggregate about 1.6 million people all over the world, including about 350,000 non-European ancestry individuals. So here, you will expect that most of the genetic association with lipid traits will have been found. So recently, we decided to aggregate data from only African ancestry individuals. This was a work that was done by a postdoc of my group, Abraham Kamisa, using meta-analysis of African ancestry individual in about 125,000 individuals. We identified 4 novel loci. We also applied the multivariate GWAS and identified additional 14 novel loci. With file mapping, multi-string file mapping yielded about 18% mean reduction in the 99% credible size set compared to the single trait file mapping. So this work has now recently published this month in Nature Communication. What I want to emphasize here is that, like I mentioned, we identified genetic variants that would not have been identified even with the largest ever GWAS in European ancestry individual because of some genetic variants that are very specific to African population. So this is also one work that we did that was led by a postdoc of my group, where we assessed lipid traits and type 2 diabetes risks in African ancestry individuals. So here, our finding was consistent with European ancestry. This is a Mendelian randomization analysis. We found that HGL exerts a protective effect on type 2 diabetes liability. So this was expected. We also find that HMG-CR inhibitor increases type 2 diabetes liability in African ancestry individuals. So, however, in contrast to the European ancestry individuals' findings, so we found that LDL may increase type 2 diabetes liability in African ancestry individuals. Again, so this raises the possibility of ancestry differences in the metabolic effects of lipid traits in type 2 diabetes. So we have been working to see whether we can use genetic to identify subgroup of individuals at high risk of complex disease. So here is some genetic risk score analysis that we did. On the left here are the discovery base. So we have data from multi-ancestry meta-analysis that include about 1.4 million people from African-American individual that has a sample from 56,000 people. You can see how much smaller this is compared to the modern ancestry from European ancestry. So what we did was to derive genetic risk score from this different discovery data set. We tested this genetic risk score on South African population and the validation in another African population. This table just to show again what we did. I want to emphasize that if you look at the on here where I put my mouse, looking at the R square for the multi-ancestry African American and European PRS, you see that the R square for the multi-ancestry was 0.69. So we had a higher R square of 1.11% from African American in the training data set. And also in the validation, we have also 2.96. You see the African American derived PRS was performing much more better compared to the PRS that is derived from the multi-ancestry, even though those are very, very large data set and the European ancestry individuals. So we decided to evaluate the transferability of PRS in different individual African population. Here, so the bar plot here shows the transferability of African American derived PRS in green, multi-ancestry PRS in yellow, in African country of Kenya, Ghana, Nigeria, from the Adam study. So what you will notice from this plot is that the African American PRS was transferable in our country compared to the multi-ancestry that was less predictive in East Africa. This is very important because the story in the literature before then was that the multi-ancestry PRS was much more predictive to all population before our study. So again, we showed that the model with the conventional risk factor of age, BMI, and 5-PCA and sex had an area under the curve, AUC of 67.9%. So while that of the African American PRS with 5-PC, age, BMI, and sex was 69.8%. So what this means is that there's an improved discriminatory ability of 1.9% if you had the PRS to the conventional risk factor. So this work was published last year in Diabetes Care. So we did a very similar thing to that work. Also, here we tested performance of PRS of lipid traits. The one I just mentioned was type 2 diabetes. What you will see here is that we showed that the African ancestry derived PRS enhanced prediction of lipid trait in South Africa population. We saw that the best performing PRS in LDL achieving R-square of 8.14%, 8.1.4%. For all the lipid traits using the African derived PRS, we were achieving R-square from either 0.87% to 8.1%. So we noted that our PRS performance was much higher than the performance reported previously in literature. So we're very excited with that result that we found we are able to transfer to South African population. So now we're excited that we're trying to do the same in another African population, this time around in Uganda population. Unfortunately, our genetic risk score prediction vary greatly between South Africa and Uganda. An example, the LDL, we had R-square of 8%. In South Africa, but Uganda, we are having R-square of 0.026%, which was very, very different. So we noted that these differences may be due to many, many factors, genetic factor, environmental factor, so many other factors, maybe diet and other factors that might be responsible. So we also noted that the South African population was from the urban population, and the Uganda population is from a rural population. So we say, so this is a comparative performance of the PRS of total cholesterol using the same genetic risk score, comparing about 286 SNPs from Uganda compared to South Africa in Zulu population. Very, very different. So this work again was published last year in Nature Medicine. So what that means is that to achieve accurate genetic risk prediction and transferability across all population, there is a need for large-scale African data set. They are very, very necessary for PRS to achieve its full potential. And the African data sets that are coming up should not be neglected in such. So we've been leveraging local resources at the MRC Uganda. I've been talking to you about the general population cohorts where we've been collecting our sample. We also have a biobank at the units where we test our sample, where we keep our sample. We also have a center where we do the biomarker testing for different kind of phenotype that's of interest. We have the Uganda Medical Informatics Center where our genetic data are stored and analyzed. So recently, we are establishing an initiative that is we call Kidney Gen Africa. This is a partnership to deliver research and training excellence in genomics of kidney disease. What we're doing is to aggregate data all across Africa, from East Africa, trying to aggregate all these cohorts, because they are all on their own small. So we want to put them together to have a large-scale GWAS done in Africa for kidney disease, and hope to do a Pan-African meta-analysis, and also try to find the effect of conditioning for ApoL1, which we know to be a key gene for kidney disease. So 2021, we started by publishing the first serum creatinine-based EGFR GWAS in Africa. So in that paper, we identified and validated 2 EGFR loci, one at a GATM locus and the second at HBB locus. So we identified those 2 and were able to validate that. So at GNTM locus, we found a distinct locus that has not been previously reported. So after doing that work, there was a paper that mentioned the need to use a much more accurate biomarker testing for kidney disease, and a biomarker testing called cystatin-C was recommended. So we again went back to the cohort and tested all the individuals for cystatin-C and did a GWAS analysis on cystatin-C. So recently, we were able to publish this paper in eBiomedicine last month, where we identify African-specific 2 novel loci that are shared with a kidney function. So our findings in this study highlight that using alternative biomarker such as cystatin-C in GWAS of kidney function can enable discovery of new genetic loci that have not been discovered or known to kidney function. So in this study, we try to assess the causal effects of lipid trace on kidney function. What you will see here is that our finding mostly supports a causal association between lipid trace and kidney function in continent Africa, also very consistent with what you will see in European population. However, we find a surprising result. For example, we find that LDL and total cholesterol, we find that causal association with higher EGFR. So this might imply that the association between LDL and total cholesterol may be non-linear. There might be a non-linear relationship between both, but there's a need for a bigger African data set necessary to investigate this. So collectively, our discovery, genetic risk score analysis, Mendelian randomization study, they show a need for improved representation of African in genomic study in order to ensure generalization of findings for precision medicine. So I mentioned that I was leading some initiatives in Africa, including Uganda Genome Resource, which I mentioned. Here is another initiative, which is what we call the Nigerian 100,000 Genome Project. So in order to improve representation of Africa in genomics, so we must create more initiatives. So here is a private public partnership with a biotech company in Nigeria and aiming to recruit and sequence about 100,000 genome in Africa. So this study, we had a consortium that is called the NCD GHS Consortium, and we have a 5-point agenda, collect sample in those 100,000 people, which we have done and completed, and biomark, create a custom array, and do some genomic study, and also empower African genomic scientists. So that's the agenda that we have for 5 years. So this agenda was published in Nature Genetics in just last year on how we plan to achieve that. So also recently we published this paper, which is just a part of the 100,000 Genome Project Plan Nigeria. This is just the analysis of about 400 samples that came from 47 self-reported ethnic group from Nigeria. Also, I like the fine-scale population structure and how we catalog novel genetic variation to Nigeria and diverse cohorts using new insight into genomic variation in understudied population. So this was published just recently, this last week. So, so the last one I want to mention very briefly is the Malawi genome resource. Here I've mentioned that this is a brand new initiative. We are just currently genotyped about 7,000 people, which we did in Africa. So this is very exciting. Well, this genotyping of 7,000 people in Africa is one of the largest ever that will genotype in the region in Africa. So the Malawi genome resource, I mentioned there are a range of NCDs, communicable disease phenotype that have been collected. There's a plan to do more with funding available. So this resource is interesting because everything is being done in Africa. They study the science, sample recruitment, the storage, biobanking, the biomarker testing, DNA extraction, the first large human genome genotyping in continental Africa, analysis and African-led. Very interesting initiative. So as I conclude, I'd like to mention that it is not only Africa that is underrepresented in genomics. Like you might know, there are two broad groups of underrepresented population. So I've mentioned the African population and residents of low and middle income countries, LMIC. There are also some indigenous and minority groups across the globe. An example, in that paper that I mentioned, we mentioned the Tiwin Island Aboriginal population in Australia, and this population that's also underrepresented in genomics. in the low middle income country. There are also many other underrepresented population, like the South Asian population, which is also underrepresented after Africa. So again, there's need to emphasize that we are doing ourselves harms if genomic study is not diversified to include all populations and capture all genetic variation. So in that paper, so we just showcase a roadmap to increase genetic diversity. So we emphasize that there's a need to leverage established infrastructure in across the globe, particularly in those underrepresented population, and also leverage the expertise and leadership within the local institution in those countries. So we also advocate for funding to maintain the existing resources within those settings, and also build capacity. Also important for underrepresented population is partnership with a global consortium and private organization. So in these days, genomics needs to be done as a collaboration, and there's need for underrepresented population to partner with a consortium all over the world, and also think beyond the box. Think about how you can partner with private organization. So in conclusion, Africa has the greatest genetic diversity. I've already emphasized that, and as such, African genomic data and resources are key, very crucial, and vital for precision medicine. I've shown today that despite some notable effort across Africa, representation of non-European ancestry group in genetic research remains very, very low. And this affects diverse global population. I show you today that from a small-sized African genome resource, about 6,000 people, we show great value for novel genetic association, discovery, and as well as for genetic risk prediction. So finally, it is to the benefit of all of us if genomic study is diversified to include Africans and as well as other underrepresented population in the world. So there's no population that should be left behind. So thank you very much for listening. I would like to acknowledge members of my group who have done most of the work that I've presented today. And I also like to acknowledge my funder, particularly Welcome and NIH for funding my project. Thank you.
Segun, thank you so much. That was, an incredible sort of journey you took us on through the initiatives you're progressing. And you do have a quite extraordinary and a little bit intimidating number of top-tier journal publications, but we won't go into that. Look, there are a number of questions that I'd be interested in posing to you. I might start with the last point you raised about the importance of collaboration in order to progress sort of representative genomics, of this fashion. And particularly the example you gave regarding the sort of genetic variant, reducing HIV load, and the potential increasing interest in of pharma companies in engaging with these genomic initiatives in Africa. You also mentioned the need for strong engagement and community involvement in prioritisation research. I'm interested in your perspectives on community sentiment about partnering with industry and the importance of ensuring benefit sharing. If pharma is developing new therapeutics, how do we ensure that the people, upon whom these new discoveries were developed can actually share in the benefit of them?
Yeah, that's a great question. I think community engagement is very key to genomic study. I always say that it's not good to just show up and say, I want to put a sample. And that has been a practice for many years. I didn't talk today about helicopter science. Maybe one of the reasons why we are where we are today is probably because in the past, there has been so many exploitation in Africa and also other low-income countries, where people from all over just fly to the country or to the continent, take sample and go back to their country without any consideration to people that that data was collected and other. So that's been there, and there are also a lot of improvement in these days, particularly in the last 10 years, with the initiative called East Africa. So there's a need for strong engagement with the community. The reason why that is important is because we need to build trust. You need to build trust with the community. We need to also, the agenda has to be created with the community. They need to understand why it is important. They need to understand that this is for the benefit of them, maybe not directly to an individual, but to the community, to the global community. They need to understand that. So if there is a strong engagement, people are going to participate in study. And I showed an example from Uganda. We have been very, very successful years going back to the same community, other community, and people are showing that. So I agree with you. So these days, there's no need to, we cannot rely only on the funding agency like NIH, Welcome. We need to go beyond that. We need to think about industry partnership, biotech company. And again, keyword here is transparency. The biotech company needs to be transparent in terms of what they bring to the table and their potential benefits in the study. So I've been having a lot of discussion with many of them in the last few years, and the same word is we need to be transparent and know if your aim is just for profit, that is not good for us. It's not good for anyone. I agree with you. There's a need for benefit sharing, and that also needs to be very clear. If you think that as a biotech company, you're going to make millions of US dollars, and you're going to give a token just for them to keep quiet, that is not the way to go. So the beneficiary has to be clear also and transparent.
All right, thank you for your comments there. It was slide 15, you had the map of Africa and the growing genomic resources being developed in each nation. There were like 20 odd little dots. Are there any plans perhaps through H3 Africa to federate these genomic resources and permit initial or additional discovery and analysis across these data sets?
Also, I'm not sure whether there's the East Africa coordinated plan, but I know there are some individual plans. There are some groups that are working on federation of some data, or maybe particularly making data accessible to global population without the data leaving the continent. So there are some initiatives. But I agree with you, it would be good to have like a well-coordinated efforts so that this data can be federated and it can also be accessible to the global population. So that would be great. So there are initiatives coming up. There's an initiative called GENCO, Center of Excellence for Genomics in Africa. I think this will fit in very well within GENCO, where data is federated within Africa and made available to global population.
Because that will be so important, not only to enrich research globally, but also for clinical care of, Africans who live all over the world.
Yeah.
All right. Oh, we've actually got a question from Loic Yengo. You had his paper up earlier. Thank you for the amazing talk. African ancestry populations have complex population structures, making inference from genetic studies quite challenging. Some of these challenges can be solved by using family-based designs. Do you see family-based recruitment being prioritised across large biobank efforts you're involved with? If not, what would be the challenges? Thank you.
That's a great question. Yeah, I think I agree completely. There's a need for family-based study being prioritized. I've seen some group focusing on this, but these days, many people are focusing on doing genome association study. Yeah. So that's where I will answer. But there are some efforts focusing on the family-based study in Africa.
Excellent. We have another question. Great effort, Segun. How would you evaluate the willingness and priority of African governments to fund genomic research?
Okay. Great. I was expecting this question, and I always talk about this most of the time. I'm always sorry to say that many African governments are irresponsible. And the reason is because they don't see value in genomics. Not because I don't blame them for that. It might be because we as scientists, we've not learned enough on how to communicate with the politician for them to see economic value. So when we get to a point where they can see economic value in what we do, I know at that point they are going to be willing to fund the research. So I think that's the only way to go. The African scientists cannot continue to rely on money from America, from the UK, and from all over the world. So African government needs to fund research in Africa, and that time will come.
Actually, that segues very nicely into Radistina's question, asking about sort of encouraging members of the population to be involved in such research, and wondering if there's any educational component to increase genomic literacy in the process of consenting people to research. But similarly, it is increasing the genomic literacy, both of our sort of governments, our politicians, as well as the public, I guess.
Yeah, that's a great question. And the way we do that is starting with the community engagement. When you go to community, so you start with some literacy, let people understand. And that's been increasingly difficult in Africa. One of the reasons why it's difficult is because I've mentioned that Africa speaks more than 2,000 different languages. So that means that study from local community to national community are going to be different. So, and also within those languages, there are a word like a DNA, like genomics, genetics, are not directly translated to local languages. So sometimes you may not be speaking, you may not understand anything you are talking about. So you must find a way to communicate what is necessary for them. But there are efforts to ensure that people understand what we are doing. And the same thing also, we start going to like in the clinic, even for medical doctors to understand why this is important and why we have to be ready for genomic medicine. And the same way, this has to go to the government. So I think most of the blame is now on us, scientists, to be able to communicate what we do clearly to participants, to other practitioners, and as well as the government agency in Africa.
Excellent. Thank you. Look, we are at time. I think we could keep talking for a while, but thank you so much for sharing this amazing body of work. And we will certainly follow your publications and the progress of your efforts moving forward. Thanks again.
Thank you very much. Thanks for having me.
So the next DNA Dialogue seminar will be on Thursday, the 26th of October, and will feature the lovely Gemma Chandratilaki of the United Kingdom. So she works with the NHS Genomic Medicine Service, and will be speaking to the systemic adoption of genomics in a national health service. So I hope you can join us then. Thanks very much.